Okay, hello people. So this is a carbohydrate anabolism. Anabolism is the opposite of catabolism, so we're going to go from small molecules to big molecules. And you should be able to, we're going to go through a couple processes, gluconeogenesis, glycogen synthesis, and the Cori cycle. You should be able to state the cellular locations for those. Describe what are called the workarounds for gluconeogenesis. Draw the substrates for the three irreversibles and state the number of carbons. State the enzymes that go with gluconeogenesis and the overall equation. Gluconeogenesis is the opposite, quasi-opposite of glycolysis. And then we'll do glycogen synthesis, and we'll also do the Cori cycle. Okay, so the question to start with is, can glycolysis run the other way? Can we make glucose from pyruvate? The answer is sort of, but with some modifications. The pathway that's the reverse of glycolysis is called gluconeogenesis. And for whatever reason, people have a really hard time remembering this word, but the pathway describes exactly what it does. So we'll break it into parts. Okay, so if you read this backwards, it says Genesis is to make, neo is new, and gluc is glucose. So if you read it backwards, it literally says make a new glucose. So gluconeogenesis is the opposite, almost exact, but not quite opposite of glycolysis. Liver cells can do gluconeogenesis, and it helps to reestablish your blood glucose levels. So if you've got low blood glucose levels, you can add more glucose to the bloodstream by the liver running gluconeogenesis. Now the trick is that we have to get around the three irreversible steps in glycolysis. So if you remember, glycolysis was glucose, and there's the first irreversible to G6P. I'm going to do the super basic version of this. And then it's isomerized to F6P. And then we have the second irreversible to F16BP. And then many, many steps. And then the third irreversible is PEP to pyruvate. So that's glycolysis. There are three irreversible steps, and each of these had a large negative delta G. So we cannot run glycolysis exactly in reverse because we have irreversible steps. So gluconeogenesis, the main points of gluconeogenesis is we're going to go from pyruvate all the way back to glucose, and we have to be able to work around, these are called workarounds, the irreversible step. So that's the most important part about gluconeogenesis, is to figure out how do we get around the irreversibles of glycolysis so that this pathway goes in a singular direction, which is from pyruvate up into glucose. So what I'm going to do is I'm going to try and draw a schematic of this. In the past, I've tried to do like individual pictures and have you try and put them together. And because I can draw on this program, I'm going to try and draw it all out for you. It's going to take a little bit of time, but I think it'll be worth it. Okay, so I'm actually going to have two cells this time with, let's just call it the bloodstream in between. So this is going to be muscle cell, and this is going to be a liver cell, and then as usual we've got our cytosol and we've got our mitochondria. So this is the cytosol, this is the matrix, this is the cytosol here. Now throughout this I'm going to be using up a whole bunch of space in the cytosol. So I'm going to have to be continually zooming in. So the bigger the piece of paper that you have to draw on, the better. Okay, so let's kind of recap. So in the muscle cell, you can take glucose to pyruvate, and that's called glycolysis. And then it can be fermented, into lactic acid, also known as lactate. Okay, and I'm going to leave out all the coenzymes and ATPs for now. Actually, maybe I'll throw a couple in there. Yeah, I should just do that. It would be good for you. NADH comes out, ATP comes out, and then fermentation regenerates NAD+. Okay, goes there. All right, when lactic acid builds up too much in the cell, that can be bad because it's acidic, and so it can lower the pH. And eventually the lactic acid will be shuttled out of the muscle cell, so you don't have a dramatic drop in the pH due to lactic acid. And where it goes after it goes into the bloodstream is it's going to go back to the liver. And we can, again, the fermentation step, if you notice, is reversible. So this is catalyzed by lactate dehydrogenase. Oops, LDH. Here we go. And in the liver cell, we go from lactic acid to pyruvate. And since this is the opposite, we're getting out NADH here. So we're running it from lactic acid to pyruvate and not the other way around. And so from here forward, I'm going to draw the chemical structures of these because I think it helps to remember. So pyruvate is here. Okay, so we're back at pyruvate, and now we're going to make glucose. And the very first going backwards through, or going through gluconeogenesis, which is us trying to go backwards through glycolysis, we have to get around the pyruvate kinase step. And this is what's called a two-step workaround. Okay, so we have to get around the PEP to pyruvate step going in the opposite direction. So the first thing that happens is that this pyruvate is going to come into the mitochondrial matrix. And again, structure. Okay, so again, we have a two-step workaround for pyruvate kinase, where pyruvate is first made into oxaloacetate. And if you remember, pyruvate has 3 carbons. Oxaloacetate has 4. So that means that we're adding a bicarbonate, which is sort of the equivalent of a CO2. So we need to add a bicarbonate here because we're going to get an extra carbon added on. So here's the carbons from pyruvate. Oxaloacetate just has an extra carbon, and it's in the form of a carboxylate. Now, in order to do this, it's going to cost some energy, and it's going to cost an ATP in order to smush that HCO3, or really it's a CO2, onto pyruvate to make oxaloacetate. You know this as OAA, one of the intermediates in the TCA cycle. It's going to cost us an ATP. So we're going to hydrolyze, use the energy from ATP hydrolysis to ADP plus PI to smush that HCO3 or bicarbonate on there. And this step requires a coenzyme called biotin. And this comes from vitamin B7. And I do have a picture for you for that, but we'll come back to it later. The enzyme that catalyzes this step is called pyruvate carboxylase. Because pyruvate is its substrate and it carboxylizes it or adds a CO2. So remember, a lot of our enzymes are named for their first part is for their substrate, second part is for what they do. Okay, so now we have OAA in the mitochondrial matrix and it will be shipped out. And we'll talk about why it is that we have this very odd step that happens in the mitochondria in just a second. But first I want to get this OAA out. I'm just going to redraw it as OAA. So now our OAA is back in the cytosol. So gluconeogenesis takes place in the cytosol and in the mitochondrial matrix. And from OAA, we can get to PEP. I'm going to do my phosphate with the circle on it. And this is going to cost us a GTP to get a phosphate on there. So you notice that OAA doesn't have a phosphate. So the phosphate here is coming from a GTP. So it will be hydrolyzed into GDP. And again, OAA has 4 carbons. PEP here has 3 carbons. So we lose a CO2 in this process. And the enzyme that catalyzes this is called PEP carboxy. Oh, that looks terrible. Yikes, go back. PEP carboxykinase. Wow, I can't spell. Carboxykinase. Okay, so those 2 steps going from pyruvate to oxaloacetate, oxaloacetate to PEP, it's already cost us 2 ATP equivalents, and that is the workaround for the pyruvate kinase step. So that's how we get around the irreversible, and you notice that both of those steps are irreversible. Okay, then from PEP, all the steps, how do I want to do this? All the irreversible steps are the same between glycolysis and gluconeogenesis. So all those irreversible intermediates are exactly the same. So what we get out is it's going to cost us, what colors am I using? Green? It's going to cost us NADH to run this, because we're going in the opposite direction here, and it's going to cost us ATP. Okay, so those are just the same irreversibles, we're just running them in the reverse direction. And so we get back to our second irreversible, which starts with F16BP. Okay, and to get around the pyruvate kinase step, there's a different enzyme, and it's called F16 bisphosphatase. Okay, and what it uses is a water molecule, and I'm actually going to draw this mechanism out for you because it's very, very simple. It uses a water to pop that phosphate off. Okay, so what goes in here is water, and what comes out is just the pyrophosphate. And that's how we get back to F6P. So we popped the phosphate off of the carbon number 1, and now we still have phosphate on carbon number 6. And I will just draw F6P here. And then it's isomerization to G6P. Again, that's the exact same enzyme that's in glycolysis. And then to get all the way back to glucose, it's the exact same mechanism as F16BP-tase. So again, a water molecule goes in to pop off the phosphate, and what comes off is the pyrophosphate. And maybe you've guessed it, but this enzyme is called G6P, or glucose 6-phosphatase. Okay, and now we've gotten all the way back to glucose, and this glucose can leave and go back into the bloodstream. okay, to replenish the blood glucose levels. If the muscle cell needs that glucose, it can re-enter. Okay, so there's a whole bunch of things going on here, but the first one that I want to point out is what's called the Cori cycle. Cori cycle. Okay, the Cori cycle, there's really no good place to say where it starts or ends because it's a cycle, but maybe I'll go with red here. I'll start with this glucose here. So the Cori cycle is basically glucose to pyruvate to lactic acid. And then lactic acid leaves and goes into a liver cell. And we go from lactic acid to pyruvate. And then pyruvate all the way back to glucose. And that glucose leaves. So it's the same starting and ending pieces, they just go in opposite directions. So in the muscle cell, we have glycolysis going on, and in the liver cell, we have gluconeogenesis. Okay. All right, so in the first workaround, the first part of the first workaround, where we go from pyruvate to oxaloacetate, in this one they're using CO2 instead of HCO3. They're essentially the exact same thing. That pyruvate carboxylase enzyme uses a vitamin called biotin. And here's the structure of biotin. It's just a structure that you need to be able to recognize. Okay. Biotin is linked to the enzyme through a lysine residue, and hopefully you recognize that it's linked through an amide bond. That's a functional group that you need to know. All right, so biotin is a CO2 carrier. So what it does in the active site, so here's the active site right here, is it acts as a swinging arm, and this pyruvate carboxylase enzyme is really quite large, but essentially what's happening is it grabs the CO2 from one region and then swings to another region to facilitate carboxylation or adding a CO2 to the pyruvate. Okay, so it's a really long group that's able to reach across sites when it moves. Okay, so it acts as a swinging arm. Do you remember what other prosthetic group we called was a swinging arm? There's this one other prosthetic group that would pick up some carbons from one site and then swing over to another subunit and drop them off. And it wasn't the heme because that one doesn't move. NADH and FADH2 usually come in and out. TPP is would be a decent choice because it's at least involved in the same enzyme. But if you remember, lipoic acid sits at E2 of the PDC, which is right in between E1 and E3, and it's able to pick up carbons from E1 and then eventually swing back over to E3. Anyways, just trying to make some connections from before. Okay, so recap of gluconeogenesis. So you'll see some figures that are like this that have both glycolysis and gluconeogenesis, and so the overall is glycolysis is here and gluconeogenesis is here. And again, in order for each pathway to proceed in what would be a forward direction for it, they have to have unique irreversibles. Okay, each pathway has to have unique irreversibles. So if we look at glycolysis, it has hexokinase, PFK, phosphofructokinase, and pyruvate kinase. That's what keeps, and we've talked about the chemical logic for this, that's what keeps this pathway moving in a singular direction. Gluconeogenesis, which starts with pyruvate and ends with glucose, has to go in its singular direction and has to have its own unique irreversible steps. So we start down here and we move up. So there's actually 2 irreversible steps that are the workaround for pyruvate kinase. So you start with pyruvate, you get to oxaloacetate, and this happens in the mitochondrial matrix. Oxaloacetate is shipped back out to get you back to phosphoenolpyruvate, and this costs you a whole bunch of energy. So if you'll notice, all the amounts of things are accounted for because we need 2 pyruvates to get to 1 glucose. So everything is doubled until you get to this point. So everything in between all this area is times 2. Okay, and then you'll notice that all of the irreversibles that are on the in-between here, they're all the same between glycolysis and gluconeogenesis. They're all the exact same intermediates. Obviously, we're going in opposite directions, so it's costing us even more ATP to get back and some NADH to get back to fructose 1,6-bisphosphate. The workaround for PFK is catalyzed by fructose 1,6-bisphosphatase, and we get a PI out of it. And the workaround for hexokinase is glucose 6-phosphatase, and we get a PI. So you'll notice that these 2 bottom steps right here, in glycolysis, they used ATP, and you would hope that in gluconeogenesis you'd get that ATP back out, but you don't. You get gypped even further. You just get some PIs, or what's called inorganic phosphate. That little I stands for inorganic. Anyways, that's the recap of gluconeogenesis. You have to work around the 3 irreversibles, and it actually takes you 4 steps to do that, each with unique enzymes. So we have an extra enzyme and an extra step. And the reason that this step is in the mitochondria is for regulation. So if you separate the 2 pathways, you put them in physically separate compartments, you're able to have better regulation of the 2 so that they don't happen at exactly the same time and exactly the same place. Because gluconeogenesis and glycolysis, you don't want them on at the same time because that's called a futile cycle. It's just going around and around and around again. It's actually overall costing you a bunch of energy. So having one enzyme in a physically separate location from the rest of them is for regulatory purposes. Okay, gluconeogenesis is super expensive. If you remember that glycolysis, we had a net 2 ATPs and 2 NADH. That's what we got out of glycolysis. Gluconeogenesis is a little bit different because of that extra step. So we start with 2 pyruvates, and I'll just do this really big arrow, and we're going to eventually end with 1 glucose. Okay, so 6 carbons here, 3 carbons there. It's not a C, that's a C. 3 times 2 is 6. Okay, to get there, it's going to cost us 4 ATPs, 2 GTPs, 2 NADHs, water, 2 waters, and some, I think it's 2 H+. That seems about right. Okay, so it's going to cost us way more than what we got out of glycolysis. And of course, on the flip side of this is what they're all broken down into: ADP, GDP, and then all the inorganic phosphates are clumped together instead of being individual. Okay, so the key here is that it's super expensive, and you only run this when you have excess energy. Okay, you only run this when you have excess energy. and your blood glucose level, and let's say, and or blood glucose is low. Okay, so gluconeogenesis is not the exact opposite of glycolysis. It couldn't possibly be the exact opposite, but it will get you back from pyruvate to glucose. Okay, so the next thing that we're going to do is, if you don't need to restore your blood glucose, but instead you need to store some of the energy or replenish the glycogen storage, we're going to make glycogen. So I'm going to redraw. I had initially thought that I would just draw everything on that singular diagram, but it's already really cluttered. So I'm going to start by redrawing, and then we'll add in glycogen synthesis. Okay, redraw, new piece of paper. I guess I'm really into yellow today, so let's stick with the yellow. Blood, muscle, liver. I'm going to make the mitochondria way tiny over here, a little tiny amount of space. Okay, so again, we'll put the Cori cycle in here in a way that maybe it makes it easier to see. Glucose to pyruvate, pyruvate to lactic acid. A buildup of too much lactic acid causes it to move out of the muscle, and eventually it will find its way into the liver. La is lactic acid, which will be oxidized into pyruvate. This pyruvate, these little squigglies are membrane proteins. Did you know that? Man, they look bad. Into the mitochondrial matrix, where an HCO3, a bicarbonate, is added onto it to make oxaloacetate. I'm not adding in ATP or the coenzymes right now because we just did that. OAA can be moved back out, be made into PEP, and we lose a CO2 here. And then there's a whole bunch of steps where we go back to F16BP, F6P, G6P, and then glucose can leave. Okay, that's the Cori cycle with glycolysis and gluconeogenesis and some fermentation. If you don't need to restore your blood glucose, and instead you need to store some of the energy and replenish your glycogen, we'll start here with G6P for glycogen synthesis. Okay, and the first thing that happens in glycogen synthesis is that this G6P is isomerized to G1P. So that phosphate is moved to carbon number 1 instead of carbon number 6. And there's going to be a good reason for that. Down, down, up, down, up. So now this is an OH again. Okay, this is G1P. And from G1P, we're going to make what's called a sugar nucleotide. Okay, this is an irreversible step. And we're going to use a brand new nucleotide called UTP. And I'm going to draw this first one out so that you can see what's going on. Okay, this is going to be uracil plus the ribose group. Negative, 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 negative. Okay, so here's what's going to happen. What color should I use? Blue? Sounds good. The phosphate on G1P is going to attack the alpha phosphate on UTP. Hopefully this is UTP. Electrons go up, they come down, and we lose a pyrophosphate. So this whole group right here is going to break off. Okay, that's what supplies the energy to get what's called a sugar nucleotide, or UDP glucose. Because what we end up with is, there's your glucose. I'm not going to draw all the things. We call this UDPG, or UDP glucose. Okay, this is a sugar nucleotide. Okay, and maybe I'm just going to move over here and have a little side note about sugar nucleotides and what they mean and what they are. So UDP is a sugar nucleotide because it's part sugar and part nucleotide. And they're good for a few things. One is that sugar nucleotides have excellent leaving groups. Okay, that nucleotide part is a really good leaving group. They are metabolically irreversible. Well, I'm sure, so wow. Let's do just metabolically irreversible. So once we get to making UDP glucose, we're for sure committed to making glycogen. Yeah, so they're metabolically irreversible, and they're basically a signal for sugar or carbohydrate polymerization. So sugar nucleotides are a signal that we're going to make glycogen. They're metabolically irreversible, so we can't go backwards. And they have a really good leaving group. That nucleotide is a super, super, super good leaving group. Okay. Glycogen is made in the liver, and the liver cytosol. And I'm running out of room, so I'm going to try and erase some of my matrix so that I can add. Okay, I'm going to erase some of that. And then I'm just going to kind of reroute my mitochondria. So matrix is here. So I have a little bit more space in my cytosol. Okay, to finish up glycogen synthesis, we have two enzymes that are needed. We have an anchoring enzyme, and we have one that just continues to add and add and add and add and add. So we go from UDP glucose to initiating glycogen synthesis, okay? And the first enzyme is called glycogenin. And it has a tyrosine as its catalytic residue. Okay. Now this tyrosine, what have I been using, is going to attack the anomeric carbon, and this UDP is going to be the leaving group. So the glucose is being transferred to glycogenin. So we have let's see, how much space do I have? Never enough space. Hey, come back here. I'll make him over here. Glycogenin with my tyrosine amino acid, because glycogenin is a protein, is now attached to the anomeric carbon. Okay, and we just keep adding on sugar units through another UDP glucose. So I'll just put another UDP glucose comes around, this carbon attacks the next anomeric carbon and gets UDP to leave. And what ends up happening is after many rounds, we have a glycogenin in the middle, and it's attached to the tyrosine, and we get a total of 8 glucoses in a row here, of course. How many is that? 6, 7, 8. Okay, I think the fluffs are better. Okay, so after glycogenin, we can add 8 glucoses. Wow, that looks terrible. We can add 8 to the chain from glycogenin. After that, all extra glucoses are added by an enzyme called glycogen synthase. Okay, and it also uses UDP glucose to add glucoses on here. So we'll continue to add and add and add and add until you fill up the glucose rosette. Okay, so kind of as a recap, we start glycogen synthesis from glucose 6-phosphate. Glucose 6-phosphate is isomerized into glucose 1-phosphate. Glucose 1-phosphate is then connected to a UDP glucose. It's connected to a UAP, which becomes UDP glucose. We lose the pyrophosphate. Glycogenin initiates the mechanism here by anchoring using a tyrosine residue. It's hard to see here, but UDP is the leaving group. Okay, and then this goes on and on and on 8 times in a row until after the 8th one is added, then all the extra ones are added using a glycogen synthase. So what you get is the glycogen rosette. So you've seen this image so far. What's at the center of this is glycogenin. Okay, so glycogenin acts as a primer from which glycogen synthase can add glucose monomers. So glycogenin is here in the middle as the anchoring, and again you can get 8 glucoses to initiate off. And if you notice, I kind of just drew over it, but I'll erase this. I'm just going to zoom. Can I zoom in here? Why can't I zoom? Let me zoom. Okay, this is probably going to get really pixelated. I'm sorry about this. But you can, it's a dimer. So you can have a tyrosine at each end of this dimer. Here's the center of the dimer right here. You can initiate with that tyrosine up to 8 coming off of each end of a glycogenin dimer. And then after the initial 8, all other glucoses are added using glycogen synthase. Okay, so glycogen synthase adds to existing glycogen chains, but it cannot initiate a chain. So that's why you need glycogenin at the very center of this. And again, this is also extremely expensive because it costs 1 UTP, which is the equivalent of an ATP, for every glucose added. Very, very, very expensive. So you only make it in a time where you've got lots and lots of energy. You don't need to make any more ATP, and you've got excess sugar, and you're going to fill up your glycogen And after you filled up your glycogen, that excess sugar is going to be made into fat, which is where we're going to go in a couple videos from now. Okay, so recap of glycogen synthesis one more time. Start with glucose 6-phosphate, isomerize to glucose 1-phosphate. Then you use a UTP to create the sugar nucleotide, which is called UDPG or UDP glucose. Glycogenin, which is a protein, initiates the glycogen chain and adds up to 8 sugars using the UDP glucose. After that, glycogen synthase adds the rest of the UDP glucoses to the growing chain, and I don't know about you, but everything sounds like glick-glack, gluck-like, and feels kind of crazy. Take your time with it, breathe, meditate on it, draw it out, leave it, go get some coffee, come back. Don't get overwhelmed. It will be okay. We'll draw it out in class, and I know that you'll get it and you'll do great. I'll see you in class.